Hi, my name is Georgios Kitsios, and I'm a physician scientist in pulmonary and critical care medicine at the University of Pittsburgh. I spend my clinical time in the medical intensive care unit and my research time in the lab, trying to answer clinically relevant questions relating to host microbiota interactions in critical illness. Specifically, I'm interested in leveraging novel technologies of microbial DNA sequencing to improve causal pathogen diagnosis in pneumonia, which remains a major clinical challenge. I'm also using emerging knowledge from the microbiome field to understand the role of respiratory and gut microbiota in the genesis, evolution, and outcome of complex critical illness syndromes such as pneumonia, ARDS, and sepsis, and then to set the stage for microbiome-targeted interventions in the ICU. So I've been very fortunate to receive a 2-year Respiratory Virus Research Award from the American Lung Association. The title of the project is Lung COVID-19 Interactions with Microbiota and Host. That comes with the cute acronym LUCID-IMHO, which addresses the challenge of Ventilator-Associated Pneumonia, or VAP, in mechanically ventilated patients with COVID-19. The problem we have here is that up to 50% of patients with severe COVID-19 ARDS who require prolonged support on ventilators may develop a serious secondary VAP by hospital-acquired pathogens, which can prolong ventilatory course, increase mortality rates, and is one of the largest consumers of broad-spectrum antibiotics in the ICU. The challenge is that VAP is notoriously difficult to diagnose with conventional microbiologic culture techniques, and we do not fully understand the host microbiome interactions in the intubated respiratory tract that lead to development of VAP. In the LUCID study, we work on 2 parallel aims. Aim 1 uses rapid metagenomic sequencing of lower respiratory tract samples for improved sensitivity and timeliness in VAP diagnosis. And Aim 2 analyzes longitudinal microbiota profiles, regional host response biomarkers in the lung, and clinical variables to understand the drivers of VAP. We do so in a cohort of 110 patients with COVID-19 and serial sampling from intubation to day 10 on the ventilator. We actually recently published results of an interim analysis in Nature Communications, combining data from both COVID-19 and non-COVID etiologies of acute respiratory failure. Well, key findings from this analysis include, first, a pattern of progressive dysbiosis in both oral, lung, and gut microbial communities in mechanically ventilated patients. The lung microbiome emerged as actually the strongest predictor of clinical outcomes, in fact, outperforming both oral and gut microbiota. Now, receipt of antibiotics with anaerobic spectrum was associated with depletion of anaerobic bacteria from the respiratory and GI tract, and that correlated with worse outcomes. And finally, we derived a dysbiosis index by which patients with low microbial diversity and high pathogen abundance in the respiratory communities had worse clinical outcome, a finding we validated in COVID-19 and even after adjustment for very well-known predictors of adverse outcome on the ventilator, really highlighting independent effects of microbiota. We consider these findings particularly promising for improving diagnosis of ventilator-associated pneumonia, prognostication of acute respiratory failure, as well as defining patient subpopulations and specific targets for microbiome-based interventions in the ICU.